like to introduce our speaker. Very pleased tonight to have Dr. Michael Heien. He is in the Department of Chemistry and Biochemistry, and he's going to talk to us about addiction tonight. So can we have a big round of applause, Dr. Heien?
Thank you very much for the opportunity to speak here tonight. The title of my presentation is Addicted Mind, What the Brain is Telling Us and Why. And I'm an assistant professor at the University of Arizona. I've been here about 5 years now, and I just want to start out by telling you a little bit about neuroscience. In terms of its field, it's a very large multidisciplinary field. You have people that study molecules, in other words, individual molecules that might bind or have actions in your brain, all the way up to cognitive neuroscience, which is just someone asking you a question. In other words, that's a huge gap between the molecular systems neuroscience, where you try to understand how different systems in your brain interact with each other, and cognitive, in other words, how people perceive the world around them. That's a huge area to span. And you have a lot of different groups working on this. You have some people working in artificial intelligence, information theory, graph theory. You have abstract verbal concepts that you might interrogate to understand how you think and how you visualize the world around you. And the work my laboratory does fits in this one little slice right here, that orange slice. We study individual molecules binding to receptors and how these molecules might change in your brain in response to different stimuli in an active system. So just to give you an overview of the research I do, I ultimately develop tools and techniques in order to understand how different chemicals in your brain are changing during behavior. So personally, my lab's driven by a goal to understand the different neurochemicals in your brain in response to either pain stimuli, in response to you making a decision to come here tonight, whatever behavioral output that you have, we want to figure out what neurochemicals are changing, how they're changing, and why they're changing, and how that ultimately affects your behavioral signaling. But the talk tonight is on addiction, and we're going to talk about the chemistry of addiction. And ultimately, there are a lot of psychological factors, but in the end, addiction boils down to one thing. It boils down to molecules that you choose to put into your body having an effect at the neuronal level. They bind to receptors, they change the way you perceive the world, and they change the way your brain interacts with itself. And ultimately, the ability of repeated exposure to drug abuse induces a change in your brain that drives you to take it again. And that's the end result of all of these addictive properties and addictive signals. Smoking has been around for a long time. People still smoke. The smoking rate is about 20% of the population smokes on a regular basis. Alcohol, we're all here tonight at Magpies. I have a couple of beers in front of me for some reason. And we all like... It's a demonstration of addiction. You are absolutely right. It is a clear demonstration of addiction. And, you know, this gentleman right here is addicted to heroin. It's a very addictive substance. But in other words, addiction is something that's innate to your brain. Anyone can become addicted to something. So I wanted to talk first about brain cells, so we understand the architecture of your brain and what we're talking about. Your brain is made up of neurons. Neurons are the computational unit of the brain. You have about 100 billion of them in there. That's a lot. And they make connections with each other. When they make connections with each other, their job is to transmit information. So here we have a cell body, and these cell bodies have dendrites. And these dendrites, their job is to receive information from other cells. You can look at this cell right here. Its dendrites receive information from this cell, but it also receives information from other cells around it. When those other cells communicate with it, it integrates that information, and then it decides what to do with that information. Should I pass it on to the next cell? Should I not pass it on? Should I change something about what I'm doing right now? That's the cell's choice. And ultimately, your brain has a lot of these neurons that are connected to each other that are making these decisions on a very rapid time scale. You decided a lot of things tonight. You chose to come here. You chose perhaps to order a pizza. Maybe you're partaking in the popcorn and a beer. That's a pretty good choice, I think. And you're making that decision based upon what these cells in your brain are telling you to do based upon your past experience and your past experience here at Science Cafe. So ultimately, these cells receive information pass it along, talk to the next cell, and then they decide what to do. So these nerve cells communicate both electrically and chemically. Electrically in that they have axons that they send electrical signals down, and once that electrical signal reaches the nerve terminus, a synapse, it causes release of neurochemicals. And these neurochemicals communicate to the next cell what to do. So here in this example, we have an axon that reaches a terminal and it forms a synapse with its neighboring cells. So it's in close chemical communication with it. Once this electrical signal reaches this synapse, calcium can come into the cell and it causes release of a certain neurotransmitter. And the neurotransmitter releases depends upon the type of cell it is. Different types of cells will release different types of neurochemicals. So these chemicals are stored in little sacs called vesicles. You know, they're very tiny, about 50 to 100 nanometers. They don't contain many molecules at all, about 5 to 50,000 molecules on average. And they fuse with the membrane. Then these neurotransmitter molecules can come out of that cell, and then they can move around. And they're going to interact with different receptors, so they can bind to receptors on this postsynaptic terminal. And then this cell that it's making contact with can decide what to do with that information, whether or not it wants to pass it along. You also have to have a way to end a signal, right? If you signal to the next cell to do something, you don't want to keep that signal on forever. So there's lots of different ways to terminate, to end that communication. One of the ways you can end that communication is metabolism. You can chew up that molecule, change it to something inactive that doesn't bind to the receptors on the postsynaptic cell. That's one way to end the signal. Another way is to uptake the cell back into the presynaptic terminal. So you have transporters that are capable of taking the chemical you release, putting it back in here so that it can be repackaged into vesicles and released later on. So in other words, these presynaptic cells can recycle the molecules they just released in order to use them in the next cycle. The next time they're asked to release more neurotransmitters, they can recycle them. But ultimately, they bind to receptors here on this cell membrane and then have an effect later on down the signal. And with addiction, molecules are addictive because they look like natural molecules. They have actions in the brain that might mimic or might look like the natural molecules that your brain releases in order to have normal function. So you can look at neuronal synaptic transmission. You have release from synapses. We just talked about that. They bind to receptors. They bind to release sites. And then ultimately, these neurotransmitters get degraded or uptaken to end their signaling properties. Well, all these drug molecules, they might look like natural substrates. In other words, they look like molecules. They're going to bind to the receptors that we're talking about for molecules your body makes. They're going to bind to enzymes that might degrade them. They might bind to enzymes that upgrade them and that uptake them. In other words, they're going to have the same effects in your body that the endogenous molecule might make. Here I have a schematic of the entire nervous system. The effects we're talking about today are just going to be in the central nervous system, the brain. A lot of drugs of abuse might have effects in your peripheral nervous system. They might affect your heart rate, your digestion, but we're going to talk about today how your brain observes these signals and what your brain does to them overall. There are a lot of neurotransmitters in your body, a lot of neurochemicals whose job it is to communicate to the next cell. This slide shows 8 very common neurotransmitters and some of the things they might do. So if we look at glutamate here, it's a excitatory neurotransmitter, meaning it makes the cell that it acts upon fire. That's its job. But it's involved in a lot of different substances in your body. GABA is an inhibitory neurotransmitter. It makes the cell it acts upon not want to fire. You might have heard of a couple of these, like serotonin. It's called the mood neurotransmitter. It's involved in depression. Most drugs that treat depression act upon serotonin reuptake inhibitors. So if you change serotonin levels, you change your mood perhaps and your outlook on life. You also change perhaps your sleep cycle and your gastrointestinal tract as well. There are a lot of different neurotransmitters, and each one has a specific function. In a few weeks, you'll hear a lot about some endomorphins and other peptide-based neurotransmitters that are a very large family. But each of these have different roles in your body. We're going to focus a lot upon dopamine today, and dopamine is called the pleasure transmitter because it evokes feeling of pleasure And anything that causes an increase in dopamine in your brain is going to be something you want to repeat. In other words, if anything you do causes dopamine increase, you're going to repeat that process, and you're going to do what you can do to get dopamine to go up again, just like you did in your previous actions. I'm a chemist by training, so this previous slide, although it's cute, it really doesn't tell you the chemical structure of these molecules. And I put glutamate and GABA up here because they're the main excitatory and inhibitory neurotransmitters in your brain. And together, these make up more than 90% of the neurotransmission in your brain. So the bulk of what you experience is due to these two neurotransmitters. However, a lot of neuromodulators and other neurotransmitters act on the signaling between these two and can change what they do. And the one we're going to focus on, as I said earlier, is dopamine. But here I have the structure of a couple of other neurotransmitters, norepinephrine and epinephrine, just so you can see that perhaps dopamine can be turned into norepinephrine. You just simply add this hydroxyl group right there on top, and you can make norepinephrine from that. And then epinephrine, you take norepinephrine and you add a methyl group off of the end. In other words, these three are structurally related. They're very similar. They have different roles in your body. Norepinephrine and epinephrine are released in response to scary stimuli. Fight or flight response, right, is released as a hormone in your body to get you to do something one way or the other. You have to make a choice what you want to do. Dopamine is a neurotransmitter that's involved in your reward circuitry that'll tell you whether or not you want to continue doing what you're doing. And I put a couple drugs of abuse up here just to show you some structural similarities between dopamine and a couple drugs of abuse. This right here is amphetamine, and you could maybe look at these two and say, well, they look very similar to each other, right? So you can see the chemical similarity between the two, so you might see how they act at similar sites in your body, similar sites in your brain. Here you have dopamine. This is amphetamine. This is methamphetamine. It has an extra methyl group on the end. This means it lasts longer in your body. It also increases its neurotoxicity. So this has a significant different neurotoxicity in your body. And this is cocaine right here. This is the chemical structure for cocaine. This might be a little harder to see the structural similarities between these two, but these two bind at exactly the same position at the dopamine transporter. The only difference is cocaine can't be taken up where dopamine can. So this works to block one of the transporters that's present in your body. All right, I wanted to talk today about how you learn things. And does anyone here have a dog? I'm sure a lot of people have pets, animal lovers. All right, maybe if just a few people. But animals are really good at learning predictions, and people are good at this too. We're really good at associative learning. This is something that was discovered by Ivan Pavlov. He's a scientist. He wanted to study how dogs digested food. So he would have these feeders come in and give the dogs food all the time. He would collect saliva from them to measure this. And the feeders made the observation that the dogs would start salivating before they got the food, right? Just when they walked in the room, they would remember that, hey, this is the guy that gives me the food. They would start salivating and drooling beforehand. So the observation is if you give a dog a steak, it's going to salivate for that steak. If you ring a bell to a dog, it's not going to care, right? It's just a bell. Dog doesn't care about a bell. It has no response to the bell naturally. Now, if you ring a bell and then bring the dog a steak, over time, it's going to learn that every time I ring a bell, I'm going to get a steak. That's the observation. So, in response to that, ringing a bell, the dog starts salivating. In other words, the response to the steak is moved to the bell. So what the dog has done is learn that I don't have to salivate to the steak every time. This bell is a really good predictor of me getting a steak. It's the same way, for you every day. You might associate something in your day with another reward. You might have a friend at work that comes by for coffee every day, and when he comes by your office, you know you're going to get coffee. So you associate that friend not with perhaps your friendship, but hey, I'm going to get coffee pretty soon. That's your association with your friend. In other words, these learned associations are how your brain works to learn that certain cues predict what's going to happen in your everyday life. And these are formalized languages. The steak's called the unconditioned stimulus because you respond to it unconditionally. The bell is the conditioned stimulus because its response is dependent upon conditioning to the steak. In other words, you have to pair that association somehow. And that involves a learning mechanism. And then you have an unconditioned response, which is salivating in response to the steak, and then a conditioned response where you salivate, the dogs in this case salivate in response to the bell. And you can look at this as far as a learning curve, right? You can look at how many times you do this to the dog and how many times it starts salivating. Eventually it'll start salivating to the bell, and then you can stop and watch it unlearn it, right? If you ring a bell to a dog and don't give it any steak, eventually it's going to stop salivating to the steak. So this is a observation, right, of how animals and how different species might learn different acts. But your brain is encoding this information somehow, right? You have cells in your brain that are firing in response to this learning. And you have a reward circuit in your brain that involves dopamine. It starts here in the ventral tegmental area. So this is a part of your brain that sends projections to different parts. And these projections signal to the different parts that either you need to pay attention right now or you're going to get a reward pretty soon. So this ventral tegmental area is a area that's very rich in dopaminergic neurons, and it projects to one of the regions it projects to is the nucleus accumbens. That's involved in pleasure center. In other words, telling you that what you're receiving or what you're about to do is going to be very good for you. It goes to other areas as well, the amygdala, which is very deeply involved in emotion, and the prefrontal cortex, which is involved in planning and cognitive behaviors. But this signaling that happens tells you whether or not something good happens or not. So let's talk about this experiment. In this experiment, we have a monkey right here in a chair, and he's just going to get juice. He's getting juice squirted into his mouth. So if you look at his dopamine cells firing, here we're going to give a little visual stimulus. So some sort of sign is going to flash up on the computer screen before him. And if this sign is this guy right here, he's not going to get anything. So if you look at, this little random pattern of lines, he doesn't see any response. Now, if you give him a squirt of juice anyway, he's going to see these dopamine cells are going to fire. In other words, he's getting dopamine release in response to the juice that you're giving him. It would be like someone walking up and giving me a beer randomly right now while I'm talking. I wasn't expecting that beer. My dopamine neurons are going to fire because I got it without expecting it. Now, if you give him a different visual cue, one that has a slightly higher amount of juice associated with it or a certain probability of getting juice with it, you're going to see the cells fire a little bit to that stimulus and a little bit more to the juice. The one I want you to think about right now is this blue and red box here. With this blue and red box, it has a 100% probability of getting reward. In other words, every time he sees this, he knows he's going to get the juice. So you don't see these cells fire in response to the juice, but you see them fire in response to the reward. In other words, this is your brain's way of encoding information. And this is called a reward prediction error. And the hypothesis is the dopamine in your brain encodes this information. In other words, you don't get dopamine firing in response to getting the reward or obtaining whatever you want to attain. You get the firing in response to things that predict the reward in and of itself. And that's how you learn, right? If you don't expect anything to happen and you get a positive outcome, you get positive dopamine signaling. If you learn to associate something in your life, something in your environment around you with something positive happening, you don't get the reward when the dopamine firing when you get the reward, but you get it with a thing that predicts the good things that are going to happen to you. And then if you have these cues that tell you something good is going to happen, but nothing good happens, you're going to get a decrease in dopamine. In other words, you made a wrong prediction, right? You thought something good was going to happen, but nothing good did happen. So then you get a reward prediction error. And this will feed back. And the next time in your you'll learn that these cues don't associate that well with the reward you get. I have a snack machine next to my office, right? And the Cheez-Its always hang up every time you try to order a Cheez-It. Like you put in your 50 cents and it doesn't fall all the time. So I like Cheez-Its, but I don't try to get Cheez-Its anymore because I've learned that the chances of me getting Cheez-Its aren't that high. They're a little low, right? So my brain is not going to try for them. Sometimes I might try, and maybe I'll get a positive outcome, and then it'll tell me, hey, you should try again. Maybe they fixed the machine. If I try and don't get it, then I'll get this negative outcome, and it'll say, hey, you were right. You should lower your probability of trying again next time for this. And the reason I'm talking about this is because dopamine neurotransmission is important for you learning. It's how you learn your everyday tasks, how you learn what to do, what to decide to do every day. But lots of drugs of abuse act upon this system. So you don't need to understand everything on this. I'll just briefly go through the fact that this is a dopamine terminal, a nerve cell that releases dopamine onto a postsynaptic space. And when you get release of this dopamine, it's typically taken up by this dopamine transporter. So cocaine acts right on that dopamine transporter and stops it from taking up neurotransmitters. In other words, if you take a drug like cocaine, the dopamine, once it's released, will hang around here forever. You have no way to terminate that signal. It's going to go on for a long time. That's a very positively reinforcing thing. It tells you whatever happened was just very good and you should keep doing that. Other drugs mess with this transporter that loads dopamine into vesicles. So there is a transporter on vesicles, loads it in there so that it can be released. Some drugs act upon that, make it spill out so that you get an increase in dopamine in the extracellular space. And my main point is that a lot of drugs of abuse work on the dopamine system directly to increase that neurotransmitter concentration in the extracellular space. My lab does fast scan cyclic voltammetry. I won't go into detail with this, but we put carbon fibers into the brains of animals in order to measure the dopamine concentrations in real time during behavior. So these are the same things you make tennis rackets out of, golf clubs, high performance aircraft, and we can put one individually into a brain and then use it to measure the time course of release of dopamine on a kind of a sub-second time scale. So here you can see the release of dopamine and then the uptake of dopamine into the transporter. A couple of observations you have when you measure dopamine is it promotes cocaine sinking. So these animals were trained to take cocaine, and in this experiment, every time they approach a lever to press it to get more cocaine delivered into their body, they have an increase in dopamine. In other words, they're expecting to get the cocaine reward, so they have an increase in dopamine that goes along with them pressing the lever. In other words, that's predicting for them that they're going to have a positive reward and they're going to see it. Alternatively, in this experiment, what we did is we stimulated dopamine neurons to release dopamine, and this is them pressing the lever while we stimulated. We stimulated right here, 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 and here. In other words, if we stimulate the neurons to release dopamine, they made a choice to go and press the lever just by electrically evoking dopamine to be released in the brain. This is a way to control behavior. So this dopamine signal tells them, hey, you're going to get something good. In this context, stimulating those neurons reminded them of the craving they had for dopamine and made them go want to press the lever overall. Okay, so drugs of abuse, these learning systems we talked about, they are ways you engage in learning with the environment. This signal tells you whether or not you're going to have something good happen to you or not. All these drugs of abuse hijack or change that system. In other words, they make it so that you don't properly learn an association of that drug with something good, because every time you take that drug, they act on your body to cause the release of more dopamine. Right? If what we're talking about is me and the Cheez-Its example, I get the reward when I go to my dopamine fires when I go to the vending machine. When I get the Cheez-Its, I don't have more dopamine going. If I did, I would have some sort of positive feedback where I thought going to the vending machine would be really, really good. That's what happens with drugs of abuse. They all act directly on your brain to cause an increase in dopamine signaling. Once that increase happens, you have not just release happening with the thing that predicted it, but with the drug itself. So here in this experiment, we have dopamine IV given to the rat. So intravenously, this is a very low dose, and you can see over a very short time scale, you have a pretty significant increase. This black line is dopamine. Goes up about 200 nanomolar, and you have a few rapid signaling events. So this dopamine signaling changes not just in response to the cues, the things that predict what are going to happen in your environment, but you always have it happen to the drug itself. And this causes positive feedback in your system. So in other words, it always makes you want to take it more. The cues that predict it make it so that you also want to take it more. And I wanted to talk about the changes in your brain that happen. So neuron morphology, we talked a little bit about synapses. And here you have a dendrite that's sent out, and it might have different shapes of synapses it forms with different cells next to it. This is a little mushroom type. You have a thin type here and a little stubby guy right there. And constantly throughout your life, throughout your day, you have your brain forming new connections with cells, new synapses with different cells as you learn new things. Some of these are pruned because you might want to forget some of that information. Some of them are reinforced because it's important things for you to remember to keep going day to day. But here in this experiment, we call this plasticity. In other words, it's your brain's ability to change over time. Your brain can change over time, can change minute to minute, day to day, hour to hour. In this experiment, this is what a synapse or one of these synaptic boutons might look like just in a control animal. If you give an animal cocaine just once and then look at its synaptic bouton, it's different. So it's different in shape. Here you see in this thing it's thinner. It also has different types of receptors on it. So it's changed its morphology in response to that drug challenge once. And it'll maintain that change for a long time. These are experiments were done in rats. Rats live about 2 years. It'll last the lifetime of a rat. So you can try to extrapolate to people and think how that might work for people. If you give them chronically, in other words, you might give an animal cocaine for a few weeks or a month and then look at the synaptic boutons, you also see changes. It looks a lot like this animal here that was just given cocaine once. If you gave them chronic cocaine then waited a couple weeks, you see a huge difference between this synaptic bouton and this one. It's a lot fatter in size, so there's different communication happening between these two. And it also has different receptors on it. In other words, its communication with the cell beforehand is changed just from being exposed to cocaine. What's interesting about this right here, these changes that happen with drugs of abuse, things like cocaine, methamphetamine, is that these are things that are supposed to happen naturally. So if you put these same rats in environments where they are supposed to learn, enriched environments that they can forage around to learn how to get their food, you might see some similar changes. But in the animals that are treated with cocaine, they show the inability to change in response to the natural environment. In other words, their brains have been kind of sidetracked or altered by some of these plastic changes. I did want to talk a little bit about addiction and substance abuse. Tolerance has a chemical basis, right? If you take a drug of abuse over a certain amount of time, you can develop a tolerance to it. And that tolerance means you need more of the drug to get the same effect that you might have had earlier. You can also have cross tolerance, which means, I could take a drug and then develop tolerance to another drug, maybe in the same family or something closely related simply by taking one. Dependence means I have withdrawal systems. If I were to stop taking the drug, I would really crave it and want it. And addiction ultimately means I'm going to keep using that drug no matter what the consequences are. Even if there are negative consequences to me, my family, my financial state, I'm going to keep doing it. And all these have underlying psychological, physiological, and chemical contributions to them. So tolerance, a couple things underlie it. One of the things your body can do is develop and upregulate enzymes. So enzymes might chew apart that drug that you take. I can produce more of those so that when I take a drug, it gets chewed apart faster. So I need to take more of the drug to have the same effect that I did before. I might have my cells change. In other words, they're going to change the receptors on them, the enzymes in them that that drug acts upon, so that I might need to take more to get the same effect that I did before. And then you could also have behavioral conditioning, where your brain changes plastically so that you actually change your response, your psychological cognitive response to these drugs of abuse. I actually have a demo today. I have a demo for someone. I brought, can I have a volunteer from the audience? I need a volunteer. Levi doesn't count, right there. You yeah, can you come up here? No, but I have a $20 bill here. About 80% of the currency in the United States has cocaine on it, just as a measure. People. This one probably does too. I'm going to give you $20 right now. This is the demo. $20 right now, or if you wait a week, I'll give you $80. What are you going to take? Going to take the $80? All right. Thank you very much. Why'd you choose $80? It's more than 20. What if I said like $25 right now, though? $25 in a week. Would you take the 20 now?
Maybe, yeah.
Maybe. What's your point? What do you think your dollar amount is? Would you wait that you would wait a week for? Yeah, for you. Yeah.
For a week?
Yeah.
A week? $30.
$30. All right, that's our cut. What was your name?
Susan.
Susan. Thank you for coming up. She's cheap. She didn't cost me any money tonight because she doesn't know that I'm not that trustworthy. And that we went back a lot of slides. There's a good chance she can't get in touch with me in a week and that I won't have to pay her the $80. If you get in touch with me, I'm good for it. But, you have to follow up. So, instant gratification means a lot. If you take a child, 3-year-old kid, put them in front of a table and say, I'll give you a marshmallow now, or if you wait 5 minutes, I'll give you a bag of marshmallows. It doesn't matter what the time or what the amount of marshmallows you promise them is. They will always take the one marshmallow now as opposed to any amount of marshmallows that are valued in the future. And it's pretty interesting. And after 4 or so, they start to learn this idea of delayed gratification. In other words, marshmallows in the future can be worth a lot, right? Like in 5 minutes, I could get a bag of marshmallows. I know a bag's a lot. 5 minutes isn't that long, and you could do the computation in your head and figure it out. But below a certain age, children can't grasp this concept, and you need to have instant gratification. You want the marshmallow immediately to satiate whatever you do. So there's something about speed of reward delivery that tells you how effective it is. Right, whether or not you choose the $20 now. $20 now is pretty good. You could get a couple of beers here tonight and some pizza before you go home, or you have to wait a week for a little bit more reward. So that's a very difficult thing to do. With kids, it's been shown that their ability to have delayed gratification is correlated pretty strongly with how successful they do in life. In other words, if they choose the bag of marshmallows later on, they typically do better in life for what that's worth. So the concept is the reward rate plays a role in addiction. We could talk about heroin. It's highly addictive. It gets into your brain really fast. In other words, you have associated you taking that drug with the euphoria very quickly because you get that response in a very fast time scale. You always don't get that rapid correlation between the things that predict the drug and your euphoria immediately. Does anyone know anyone that's been addicted to Nicorette gum that wasn't addicted to cigarettes beforehand? I mean, the reason Nicorette gum exists is to wean people off of smoking cigarettes, right? The time course of nicotine going into your body is a lot slower when you chew it versus when you smoke it. When you smoke it, it's seconds to get into your bloodstream and have an effect on your brain. When you chew it, it's minutes. So if you gave teenagers nicotine chewing gum, they wouldn't get addicted to it, because their body doesn't associate the pleasurable part with the chewing that well. It's a very loose association. So what we're saying is the reward rate, how fast you get that dopamine release, matters. Nicotine, when you smoke it, gets in your body very fast. So that happens. This plays a role when you design drugs or when drugs are being used. The pharmacokinetics and pharmacodynamics of the drug are really important. What we're talking about is how fast the drug gets into your brain. Your brain has a great need for nutrients and oxygen, but it doesn't want to be in danger of having a bunch of compounds in your blood into it. So you have a blood-brain barrier. And this blood-brain barrier only allows certain molecules to pass into your brain and doesn't allow other molecules to pass into your brain. So this limits types of things you can turn into drugs and the types of things you can get into your brain in general. Your receptors are pretty large molecules, and how well these things bind to receptors and have their effect will also change, you know, the addictive properties of the drug. And you can change things to make them more addictive or not. I put together a couple of timelines for 2 drugs of abuse. And these are, you know, these aren't everything in the drug timeline, but you can think about ethanol. About 6,000 BC, people started to grow grapes to turn into wine. And they started to drink wine, maybe around 6 and 4,000 BC. Then around 3000 BC, we had a lot of civilizations making beer. And you could even have some beer recorded on clay tablets. There's a couple of companies that try to recreate these ancient beer recipes for you today to think what ancient beer looked like. And this is I think my favorite one right here. Cuneiform tablet recommends beer as a tonic for lactating women. So that's something around 2200 BC. And then you have a couple of events here. In 625, Muhammad said that you can't drink alcohol anymore. So Arabs developed caffeine after that. Coffee became popular. That's also a pretty good drug, a pretty good drink, the substitute for alcohol. But also you can see just a couple hundred years later, Arabs discovered distillation of alcohol. So he said you couldn't drink it, but they figured out a way to make it really concentrated just a couple hundred years later. And then the first in Western society, the first documentation of alcohol distillation was in 1100. It's not too long ago, if you think about it. Less than 1,000 years ago was the first time any European had distilled alcohol, and they called it spirits, as it's the extracted spirit of wine that they have in there. And then after that, this is just from England right here. We have being an English-speaking society, we have a lot of records from England. But distilled spirits first became apparent in England in the 1500s, and then they had a tax on distilled spirits because people were drinking too much. Alcohol had been tied to everything in every aspect of your life. Anything that happened was tied to alcohol, so people began drinking a lot, and then they taxed it quite a bit, and then the moonshine trade developed. 1920, prohibition of alcohol began in the United States. We're past that now. You can look at cocaine now, and it has a slightly different timeline because there's evidence of people chewing the coca plants in South America in 5000 BC. They would chew these leaves, make tea out of it, make whatever out of it. And then it wasn't until the 1400s that Europeans operated plantations to grow these leaves and to make tea, to chew it the same way that the ancient people did. In 1859, a German graduate student, Albert Niemann, isolated cocaine from these coca leaves. Right after he did that, he was able to isolate this product, and then a few years later, Merck made a 1/4 pound of cocaine from that. A few years after that, Merck produced a 3/4 pound of cocaine. Here we are, though. That's about 30 years. That's not too much, right? Sigmund Freud publishes a paper, and then he says, hey, we should give cocaine to people to treat a bunch of different stuff. And then the next year, Merck produces 3,000. The same year, Merck made 3,000 pounds of cocaine. A few years later, they're making 150,000 pounds of cocaine, and it's becoming popular and in your everyday culture. This is a little flyer of a French tonic wine. A lot of wines, some wines that were popular in Europe had cocaine added to them. The Pope endorsed this one himself, Vin Marani, because it helped keep him awake. It has a little bit of wine in it and some cocaine. I'm sure it tastes delicious. This was popular in the United States as well. Coca-Cola was introduced. Here it was called Coca-Cola. Coca from that coca right there, the coca plant. It had cocaine in it. Cola was from the plant they extracted the caffeine from. So it has both caffeine and cocaine in it, and it originally. And then in 1914, the United States outlawed stimulants, quite a few of them, such as cocaine, in the Harrison Narcotics Act, so you couldn't sell it in your soda anymore. And then 2008, though we're about 100 years past that, global cocaine consumption is about 100 billion pounds. The USA consumes about 350,000 pounds annually for a street value of about 25 billion. When you compare it to where we were 100 years ago, you can see how we might or may not be doing. One thing I'll say here is these forms that we started out in weren't addictive. People chewing coca leaves is not addictive, and it's not addictive because of what we talked about earlier. That time frame isn't really there. You don't get the drug that fast. No one that makes tea out of these leaves has become addicted to the tea. They can quit at any time and not have the issue. But if you concentrate it, yes, it's a very highly addictive drug. So we could come up with a generic timeline for all sorts of drugs that you might do. You have a discovery where you say, hey, this is really good. It's been good for people, perhaps. It makes us feel good. It's good for humanity. You might enhance it a little bit. And that could involve selective breeding, isolation of the drug, concentrating it, maybe figuring out a different way to make it. You might alter its structure to make it more potent, to make it bind to those receptors better. You then start to observe problems in your society that arise from the use of that drug. You ultimately ban that drug and say you can't use it anymore. And then we end up in the end saying, all right, you can use it, but we're just going to tax it heavily and make money off it. So this is our generic timeline for drugs of abuse. So in summary, the couple things I want you to take away from this are synaptic transmission involves neurotransmitter release, and all drugs of abuse mess with this synaptic transmitter release in some way. They change the way your nerve cells, your brain cells communicate with each other because they're changing how they talk to each other. They change your perception of the world and how you interact with it. These drug molecules often look like their natural substrates. They look like the molecules in your brain that might be acting on there. And the impact of that drug ultimately depends upon the specifics on how it binds, its interactions, molecular interactions with the receptors and enzymes, and its ability to get into your brain and how fast it gets in your brain. Those are the ultimate things that change it. I would like to thank my group here. I didn't show a lot of their research, but they're my research group, and we work on studying the brain, understanding how your nerve cells talk to each other. This presentation here was funded through NSF grant to Professor Cowan and I to develop new tools to measure brain interactions and brain chemistry. And you can follow me on Twitter if you want to at ML Heien. I think we have a question and answer period. I'll be happy to take any questions.